So today we're talking about species and speciation. Does anybody know what these organisms are? Caterpillars! And you can tell by looking at these 4 different caterpillars that they're different species. They're different organisms. They live in different habitats, and they've developed over time, they've evolved to have traits that help them survive in their particular environment. For example, this one has evolved to look like a spider, so it scares off predators. This one evolved to look like a snake, so there would be the snake eyes, to scare off predators. These two have either hairs or barbs that have toxins in them to deter predators from eating them. So different strategies have evolved in these different organisms over time to help them survive in their environment. So we had a change in allele frequency over time, and we have different caterpillar species. Now we all know they're still caterpillars, so today we're going to talk about what makes these caterpillars different species of caterpillars. So there's 2 evolutionary pathways that we need to understand when we're thinking about how different species arise. The first is microevolution, and that's evolutionary changes in a species over a relatively short period of time. So this is what we've already talked about. This is when we use Hardy-Weinberg to see if our allele frequencies are changing in a population from one generation to another. So we can just look at allele frequencies to see if this is happening. So here we have some beetles, and in our first generation of 6 beetles, 75% of the alleles were the dominant green allele, and 25% of the alleles were the recessive brown allele. In the second generation, after these 6 individuals mated, 71% of the alleles were green, and 29% were now brown. So this is an example of microevolution. Our allele frequency has changed over a generation. So micro, small, short period of time. The other type of evolution that we're interested in as biologists is macroevolution. And this is evolutionary patterns viewed over long geological scales. So if we know these are all beetles. There's many different species of beetles. Here's our one species of beetle in the red dot that we're interested in. We know many beetles there's different species of beetles, and they're all similar to each other. And we know that beetles are more similar to other insects than they are to other organisms. And then we know this is arthropods. So we know that all arthropods, so spiders, ticks, crabs, shrimp, these are all more closely related to beetles than, say, mammals are. And so that's macroevolution. So macroevolution is all of the parts of microevolution, which include mutation, gene flow, genetic drift, natural selection, plus a long period of time, 3.8 billion years, and that's how we get macroevolution. Any questions on the differences? Okay. So divergent evolution is selection that favors changes, making 2 or more groups less similar. So if we're thinking about an organism, say we have this bird, and it flies across the ocean to the Hawaiian Islands, one bird species. Over time, the birds that live on this island, the one species, will change because allele frequencies will change. There may be mutations that occur that will allow certain species to enter a new habitat or use a new resource, and they're better adapted, and so that mutation stays in the population. This can lead to speciation. So if we have our single bird that went to the Hawaiian Islands a long time ago, new habitat, no other bird species are there. There's different resources available. There's nectar from plants, there's insects, and there's seeds from plants. So this one bird species, over time we had divergent evolution, and we now have many different bird species that are adapted to either sipping nectar from certain types of flowers, getting insects, or eating seeds. And so because we have these different fitness levels for different strategies for getting food, we have divergent evolution. We now have many different bird species with many different types of beaks. Adaptive radiation is divergent evolution from a common ancestor by adapting to different parts of the environment. So it's a type of divergent evolution. Adaptive radiation is really common when organisms enter a new habitat, such as an island where there were no other organisms before. So this one is an example from the Hawaiian Islands, and the most famous one that people like to think about are when we talk about Darwin's finches. So Darwin's finches are a closely related group of 14 finch species that live on the Galapagos Islands. They have one common ancestor finch from South America, from the mainland. That's what it looks like. And over time, they evolved due to changes in allele frequencies to seed eaters. There's one bug eater, or bud eater, and then a bunch of insect eaters. And so from one species, we had adaptive radiation because different birds were better adapted to being able to get nutrients from different resources. Those organisms that were better adapted, say, for example, to catch insects, those traits stayed in that population, and over time, we created new species. Convergent evolution is kind of the opposite. So divergent evolution, we're diverging, we're differentiating, we're becoming different. Convergent evolution is selection that favors changes making two or more groups more similar. So we have placental mammals, all up here, which are organisms like us that our embryos and our offspring develop in a placenta. We have marsupials, where their offspring actually develop in a pouch. So kangaroos are the most common marsupial people like to think of. In North America, does anybody know what our marsupial is? The possums. So their offspring develop in a little pouch on their stomach. Wombats are also marsupials. They have little pouches. Cool thing about wombats is their pouch is actually facing down instead of up because they dig burrows, and so to make sure that dirt doesn't get onto their offspring in their pouches actually face towards their back feet, so when they dig, they push the dirt out and not onto their offspring. Cool marsupial. These mammals and Australian marsupials are not that closely related. We're all mammals, but that's what we have in common. And that these organisms have formed analogous structures, which are structures that perform similar functions, but not necessarily having similar ancestry. The most common one is thinking about birds and bats. Birds and bats both fly. Bats are mammals. Birds are not. But they both have wings that help them fly through the air. So similar functions, wings help you fly, but we know birds and bats have different ancestors. Same with mammals and marsupials. We have the placental mole and the marsupial mole. They both have specialized claws for digging, and they are both blind because they live underground. They do not need to see. We have climbers like the ring-tailed lemur and other lemurs that live on Madagascar, and the spotted cuscus. They have long tails and grasping hands that help them climb through trees. So even though these organisms, Madagascar is an island off the coast of Africa, Australia is its own giant continental island, very far apart, but because these organisms both want to inhabit similar habitats, over time they have developed similar characteristics or analogous structures. Biological species concept. The biological species concept is members of the same species have to interbreed in nature, so they need to mate with each other. They need to produce a viable and fertile offspring, and they cannot produce viable and fertile offspring with other species. So those are the three things that are required for the biological species concept for a group of organisms to be considered a species. So here in this picture from your textbook, we have 3 different species of butterflies, and we can see that if we're looking at structures, their wing lengths differ, and their antenna lengths differ. And so these butterflies with short wing lengths and short antenna lengths cluster together. Then we have short wing length and long antenna, and long wing length and medium antenna. Organisms that cluster together, we can typically think of as a species. To test to make sure that these organisms are different species, we can put them together and see if they will breed. If these organisms did breed, even though they look differently, if their offspring are viable, we will say they were the same species even though they look different. If we put them together and they did not breed, we would say they are two different species. So how does this occur? This occurs through speciation, which is the process in which a new species arises. And you need three things for speciation to occur. You need vicariance. Vicariance occurs when a single interbreeding population is split into two populations. And this typically occurs through some sort of environmental event. So there might be an earthquake and it splits two populations apart and there's a giant canyon in between it so they cannot meet with each other anymore. Maybe there's a flood, and after the flood, a river formed, so the two populations can't swim across the river to mate with each other. Maybe a volcano erupts. Something happens, a vicariance event in the environment that forces two populations to be split from each other. After that happens, we must have genetic isolation. So those populations are isolated, they're separated, they are not reproducing with each other. And remember, this is going to be a random event, so the allele frequencies for our two populations might be different. Over time in our populations, different mutations will occur that might be advantageous, so they might remain in the population. So no gene flow between these two populations, so that over time we'll have genetic divergence. And that means that over time, these two populations will become different from each other. Their allele frequencies will diverge. They will become very, very different. One way to test if these organisms are two different species is to bring them back together. Or if that river that formed from that flood dries up and the two species might be able to come back together naturally. When that happens, there might be things that have occurred in these two populations that make them so different from each other that they do not reproduce anymore. And so that means that these organisms will be reproductively isolated. So we're going to talk about different mechanisms for reproductive isolation. So there's two different types of reproductive isolation. There's pre-zygotic and post-zygotic. What is a zygote? It's a fertilized egg. So if we're pre-zygotic, it's before the zygote. It's before fertilization of the egg occurs. So our mechanisms of reproductive isolation are preventing gene flow between populations that have come back together. So the first kind of pre-zygotic isolation is temporal isolation. So we're going to think of time. So maybe these orange frogs like to mate during the day, and the blue frogs like to mate at night. So they're mating at different times. So they're never going to be together, even if they're together in the same place, they're not trying to mate at the same time. So they're still going to be isolated. There's no gene flow. Another population of frogs might mate in March, and another one might mate in October. So those two populations would not mate with each other. There's no gene flow because when they're ready to mate, they're different times of the year. So different times, different seasons, different times of the day. Temporal isolation. The next type of pre-zygotic isolation is habitat isolation. So if over time, the orange frogs have evolved to live in the tree, so now they're tree frogs, they're going to look for mates in the tree. If the blue frogs still live on the ground, they're going to look for mates on the ground. So because these two or populations live in different habitats, they are reproductively isolated and they will not have gene flow because the blue frogs are not going to hop up a tree to look for mates. They're going to look for mates on the ground. The orange frogs are going to look for mates in the tree, so they're isolated by the habitat that they're living in. The next is behavioral isolation, and that has to do more with what's going on with the behavior that these organisms are displaying. So the blue frogs make a certain noise to attract mates, and that's attractive to the other blue frog. But to the orange frog, they don't like that music. They're looking for a different notes. So that behavior, that mating call, is going to keep them from mating with each other. It's not going to allow gene flow to occur. Another example would be for birds that do mating dances, and they put out their plumage, the males do, that attracts females of their species. Females of other species do not find that attractive, and so they won't come to mate with those birds. Fireflies do the same thing. I know when you go outside in the summer and you see their butts lighting up, different species light their butts up in different patterns, and that's how males and females find each other. So if your lighting pattern coming out of your butt isn't attractive to a mate because it's different, they're not going to have gene flow. They're not going to mate with each other. Then we also have gametic isolation. So this is still before the zygote because we do not make an egg, but these organisms try to mate. So say our blue frogs and our orange frogs try to mate with each other, the fusion of the egg and the sperm cannot occur. So there's some sort of incompatibility between the egg and the sperm. So for example, this is just a schematic to kind of show the idea. If the blue sperm and egg use a mechanism that is a square shape to recognize each other, and the orange sperm and egg use a triangle shape to recognize each other, when the blue and orange sperm and egg come together, they can't recognize each other. The sperm will not be able to enter the egg. We will not have a zygote. So there's something happening with the gametes that they cannot fuse together. And the last type of prezygotic isolation is mechanical isolation. So there's something wrong with the genitalia of the organisms that do not allow them to actually copulate or have sex. A really classic example are in these organisms, which are damselflies, related to dragonflies, and these are different structures of the male penises of copulatory organs of these damselflies. And you can see different species of the males have different shapes of their copulatory organs, and those only fit inside females of the same species. So because of the shape of, for mammals, the vagina and the penis, or for insects, copulatory organs, they won't fit, so there's mechanics that aren't working, and so they will not be able to reproduce even if they try. Any questions on prezygotic isolation? Yeah, you never thought you'd see insect sex organs in class, right? So we have two types of post-zygotic isolation. So this is after the zygote. So sex happened and fertilization of the egg actually occurred. What happens in post-zygotic isolation is either you have hybrid inviability. So even though your two frogs mate, they cannot actually produce an offspring that is viable. So the female will have some sort of miscarriage, or their offspring will not survive to reproductive age. So even if their offspring is born, the offspring will die. It will never be able to reproduce. It's not viable, but usually a miscarriage will occur. So no offspring will ever be produced that can live to reproductive age. The other type of post-zygotic isolation is hybrid sterility. So you have your initial frogs, they mate, and they actually produce offspring that live to reproductive age. But when these hybrids, the combination of these two frogs, try to mate, They cannot. So that occurs for groups of organisms that are not totally reproductively isolated yet. They might still attempt to have gene flow, but their offspring cannot have offspring. So this occurs when people try to mate lions and tigers together. Their offspring, they produce offspring, either tigons or ligers, depending on who is the male and who is the female, but those ligers and tigons are sterile. They cannot reproduce. Another example is when a horse and a donkey mate, they produce a mule. Mules are sterile. They cannot have offspring. So we know mules and donkeys are two separate species. We know lions and tigers are two separate species. And then just to put it all together, I put a little table in here for you. So when you're studying, you had them all together on one table with examples. So there are modes of speciation. So how do species occur? How does speciation actually occur? So there's allopatric speciation, which is speciation by geographic region. So typically in allopatric speciation, we have a vicariance event. So for example, there's a new river that changes course and splits our species. Over time, we'll have two new species. Or for some reason, there's dispersal of some of the population to a new area. And over time, they will become two new species. There's another example of allopatric speciation called peripatric speciation, and that's only when a small group of individuals starts a new population. Over time, allele frequencies change, and then you have two new species because they are reproductively isolated. Even if they come back together, they will not be able to produce viable, fertile offspring. Bless you. The other kind of speciation is sympatric speciation, and that's speciation in the same geographic place. This doesn't occur as often because we're in the same place the whole time. So we have our ancestral population, and for some reason there's disruptive selection. So remember, we're going to the two extremes of our population, and the disruptive selection is extreme enough that over time, the two extremes do not mate with each other, and you will have two new species. So I'm going to go through each of these in more detail with pictures. So allopatric speciation. New species is from geographically isolated populations. So there's something that happens. For example, we have, because I like insects, beetles, there's a river that forms. Which organisms were on either side of that river was random, and over time, mutations will occur and allele frequencies will change so that now we have two very distinct populations. So you must have geographic isolation and no more gene flow, reduction in gene flow between these two populations. A way to remember this is allo means other, patric means place. So the populations are in another place. They're separate from each other. Then we have peripatric speciation, and this is a new species formed from a small population isolated at the edge of a larger population. So peripatric, peri means near, patric means place. So the way to think about it is a little fruit fly is pregnant and it's stuck on a banana, and the banana goes to a new island. And whatever alleles this small population of fruit flies has, over time will have changes in allele frequencies, and they will now be a different species. The island flies will be different than our mainland flies. And this peripatric speciation occurs more quickly because you have a smaller starting population, and so you have more genetic drift happening. So parapatric speciation is kind of a weird but cool type of speciation. And this occurs when new species are formed from a continuously disturbed population. And we find this occurring in human-dominated landscapes where we have disturbed the environment. That's where this most typically is happening. So para means beside, patric is place. So we have uncontaminated soil and soil that is contaminated with waste from coal mines. And the original population of insects lived on the uncontaminated soil, but for some reason, some of these organisms, through random chance, random mutations, are able to actually survive and thrive on this contaminated area. Because they are relieved from competition and natural selection, because most organisms cannot live on this, they are able to survive and reproduce, and this allele that allows them, this mutated allele that allows them to live on the mutated or the mined waste, gives them an advantage here where they don't have the advantage on the uncontaminated soil. So over time, they become a new species that's able to live on mine waste. And sympatric speciation, which is new species formed within the range of the ancestral population. So we're in the same place the whole time. So sim means same, patric means place. We're in the same place. Typically, new species arrive when they find a new niche. So for example, we have green and red apples in our population. I apologize if you're colorblind. This was the best picture I could find. If certain flies like to eat and mate on the red apples, and certain flies end up switching over to the green apples and they prefer that, over time what can happen is if the red apple flies only mate with the red apple flies and the green apple flies only mate with the green apple flies, we'll have a change in allele frequency, and the red apple and the green apple flies will become two separate species. So this is how sympatric speciation occurs. So if a new plant enters an environment, certain insects can use that plant for nutrients in the same environment with their ancestral population. Over time, they could become a new species. This does not happen as often as allopatric and peripatric speciation. So another way to think about this is if we have natural selection happening, we have our number of individuals on our y-axis and bill size, the size of the beaks of birds, on our x, and we start small and we get bigger to large. Natural selection can eliminate medium-billed birds in an environment if no medium seeds are around, and bill beak is equal to the types of seeds you can have. So if there's no medium seeds for you to eat and you have a medium-sized bill, you're going to die because you're going to starve to death. If you die, you have a lower fitness because you're not surviving very long, not reproducing and having many offspring. So that trait is not being passed down to the next generation. The organisms with large beaks that can eat large seeds and with small beaks that can eat small seeds are able to survive. They reproduce, they pass on their traits for beaks, for bird beaks into the next generation. Over time, we'll have a lot of birds with small beaks and a lot of birds with large beaks. If they stop mating with each other because, say, these seeds are available at different times of the year, then we will have sympatric speciation once there's no more gene flow. So then we'll have a species with small beaks and a species with large beaks. Any questions?